So Rötig ataxia is a neurological disease with a progressive cerebellar ataxia. Patients also present cardiomyopathy and some of them also diabetes mellitus. This disease is a genetic disease, a recessive ataxia, a relatively rare disease, and the gene encoding, the gene responsible for the disease is the FXN gene that encodes the frataxin, which is, I don't know, no, it doesn't work. Can I have the pointer here? No. So I will try with the mouse. So the FXN gene encodes a small protein which is called frataxin. And this frataxin, the mutations are due to repeat expansion of the GA triplet in the first intron, and this results in an unstable transcript and a low amount of frataxin. So this is a loss of function disease. The frataxin is a mitochondrial protein. I guess that most of you know that. It's a very important protein involved in the biogenesis of iron-sulfur clusters. Here is the different proteins involved in its biogenesis, and frataxin is in the first step of this biogenesis. So it is located in the mitochondrial matrix, and it is ubiquitously expressed and very conserved among species. This iron-sulfur cluster, after the biogenesis in the mitochondria, will either be associated with mitochondrial proteins, proteins for the respiratory chain, proteins from aconitase, from the TCA cycle, and also lipoic acid synthase and ferrochelatase inside the mitochondria. And the iron-sulfur cluster also exit from the mitochondria and will be integrated, associated with a lot of different proteins in the cytosol, in the nucleus, and in different subcellular locations. So it means that frataxin defect should result in mitochondrial respiratory chain defect, which is the most known defect in this disease, but also in a lot of different proteins, probably with different level in different tissues. So we focus here our interest on the iron, and first we quantify the iron inside the mitochondria in 5 fibroblast cell lines from patients with Rötig ataxia compared to controls, and as you can see, there is an mitochondrial iron overload. We also observed a cytosolic iron overload, and the ratio between cytosol and mitochondrial iron is always the same compared to controls. So it means that we have mitochondrial and cytosolic iron overload. Therefore, we looked at the iron homeostasis, and I guess that most of you know how iron uptake is done in the cells. So iron is bound to transferrin that will bind the transferrin receptor and that will enter the cells by endocytosis, and this early endosome then will be acidified and leave the iron that will be stored in the ferritin, and the endosome go back to the membrane with the transferrin receptor and the transferrin, and to have then a new cycle of transferrin receptor endocytosis and iron release. There is another way for iron uptake for non-transferrin bound iron with the NTBI transporters, and the regulation and the function of this transporter is not so well known as for the transferrin receptor. And finally, iron will exit from the cells using the ferroportin. We looked at the homeostasis of iron in fibroblasts of these patients by incubating them in high iron concentration, either using holotransferrin that will bring iron through the transferrin-bound iron uptake, or with ferric ammonium citrate that will use this system of non-transferrin-bound iron. And you can see in both cases that control cells slightly increase the iron content, the intracellular iron content, but not so much. However, fibroblasts of Friedreich ataxia patients have a major iron overload, meaning that these cells are unable to regulate the iron uptake, and this results in a major iron overload. So maybe most of you know this, how iron homeostasis is regulated. The main regulation occurs at the post-transcriptional level. There is two iron regulatory proteins, IRP1 and IRP2, that binds to iron regulatory elements in 5' or 3' untranslated region of different genes involved in iron homeostasis. When IRP binds in the 3' region of the transferrin receptor transcript, it protects it against RNase, whereas this occurs in the low iron condition, and at the same time, this IRP binds to the IRE of ferritin in the 5' part of the transcript, hampering the translation and the synthesis of the ferritin. On the contrary, in high iron condition, IRP1 become an aconitase and lose the IRP function, and IRP2 bind to the FBXL5, which is a protein involved in the sensing of iron, and this FBXL5 bring it to the proteasome for degradation. So there is no more IRP on the IRE, therefore we will have an increase of the a decrease of transferrin receptor that will limit the iron uptake, and increase of ferritin for iron storage, and an increase of ferroportin for iron export. So this is very important, and this is very tightly regulated. So we looked at that regulation in fibroblasts of patients, and as you can see here, the transferrin receptor transcript in low or high iron concentration seems to be okay because we have relatively the same amount in control and patient cells. However, I remind you that patient cells have a very important iron accumulation. So we should have in these patient cells a much more decrease of transferrin receptor, which is not the case. Therefore, we looked at the iron regulatory proteins to iron regulatory elements binding activity. And as you can see here, either in low or high iron condition, we have an increased IRP2 to IREE binding activity. So this was known since very long time, but it was considered as a consequence of cytosolic iron depletion, despite nobody measured the iron content in these cells. And as we show here that we have a high iron overload, we can say that the frataxin defect results in increased IRP2 to IRE binding activity. And we hypothesized that this is due to the fact that FBXL5, the protein, the sensing protein of iron, is an iron sulfur cluster protein. This was shown very few years ago, and the location of the iron sulfur cluster is very close to the location of the IRP2 that binds these proteins. Therefore, due to the decreased to the iron sulfur cluster defect, we hypothesized that the binding of IRP2 to FBXL5 will decrease, whereas the binding of ARP2 to IRE will increase. And this could be the reason of this increased binding activity. We also looked at these proteins by Western blot, and as you can see here, so you have to look to, we have 1, 2, 3 controls and 5 patients. What you can see here is that in low iron condition, we have increased amount of iron in the patient cells because there is an iron overload, and also this increase in the high iron condition. Transferrin receptor here in high iron condition will decrease in control cells, but it doesn't decrease in patient cells, and it rather increase. And for the ferroportin, we should also have, where is ferroportin? Here. It increased in control cells in high iron condition, but actually it decreases in the patient cells in high iron condition. So in this Friedreich ataxia patients, we have completely abnormal regulation also at the protein level. This is the consequence of the abnormal transcription regulation that result in iron overload. We also looked at the transferrin recycling. Yeah, I guess that we can have the movie. As you can see here in the control cells, we follow the transferrin that decrease relatively rapidly in a few minutes. And in the patient cells, yeah, we have here a delayed of transferrin recycling that will release much more iron than that needed, and that will result also in iron overload. Here is a quantification of the transferrin recycling in the 3 controls and in the 5 patients. So frataxin defect results in a very important delay of transferrin recycling. What we know about transferrin receptor is that these proteins undergo palmitoylation. And palmitoylation is the protein modification by addition of the palmitate. And as you can see here, in the 5 Friedreich ataxia patients fibroblasts, we observed a very severe palmitoylation defect compared to the controls. It's less than 20% of normal palmitoylation. And this palmitoylation is a post-translational protein modification that is involved, that modify the protein trafficking, the protein stability, the protein-protein interaction. And this is a reversible mechanism that regulates the protein localization. So there is one molecule which is called artesunate that is known to induce palmitoylation of transferrin receptor, and that will result in the reduction of transferrin receptor cell surface levels. We observed accumulation of transferrin receptor at the cell levels, and we tested this molecule that could be very helpful for Friedreich ataxia. And indeed, as you can see here, artesunate partially rescue the transferrin receptor. You should not compare the 2 experiments because there is 2 exposure, but as you can see here, we really increase the palmitoylation compared to control cells. This results also in a decrease of the accumulation of transferrin receptor at the cell periphery, and also to, this is the quantification, and also to a decrease of iron content. The black bar corresponds to the cells treated with artesunate. This palmitoylation is also, I will go directly to the next slide, is also affected for the NTBI transporters, the DMT1 and ZIP14. As you can see here, DMT1 here has a low palmitoylation in the control cells compared to the patient cell compared to the control. And similarly, ZIP14 has also a low palmitoylation level. So the frataxin defect impairs the post-translational regulation of transferrin receptor, DMT1, and ZIP14. So now the question was, how can we explain that a defect of frataxin, which is an intramitochondrial protein, should affect the palmitoylation of extramitochondrial proteins, especially transferrin receptor, DMT1, and ZIP14. Actually, among the iron sulfur cluster proteins that should be affected by a defect in frataxin, here in the mitochondria, we have the lipoic acid synthase that is involved, it allows the synthesis of lipoic acid that is a cofactor of several proteins, including the pyruvate dehydrogenase. So it is well known since very long time that in iron sulfur cluster biogenesis defect, there is a decreased lipoylation of several proteins, including the pyruvate dehydrogenase, so we check that. And that this defective lipoylation is rescued by dichloroacetate, which is an inhibitor of the PDH kinase that itself is an inhibitor of pyruvate dehydrogenase. So we measure the coenzyme A content in patient cells, and as you can see here, we observed a decrease of the CoA pool in Friedreich ataxia patients. Actually, we are using a kit measuring the coenzyme A, but I guess that it also measuring the acetyl-CoA is not very specific, and this should reflect the limited acetyl-CoA pool in Friedreich ataxia. And then we complemented the cells with coenzyme A that obviously rescued the level of coenzyme A. And then by treating the cell by dichloroacetate, we increased the coenzyme A pool, showing that the frataxin defect indeed limits the coenzyme A pool. And acetyl-CoA is the sole donor of acetyl groups for acetyltransferase or palmitoyl transferase, the proteins that allows the palmitoylation. Therefore, we treated the cells of Friedreich ataxia patients with coenzyme A. This increased the transferrin receptor, and also dichloroacetate increased the transferrin receptor palmitoylation. So these two drugs, these two molecules, are therefore drugs for Friedreich ataxia treatment. Here we also measure the effect of coenzyme A, dichloroacetate, and artesunate on iron content. And as you can see here, using fibroblast incubated with holotransferrin for measuring the TBA uptake or non-FISFAC for NTBA uptake, we very significantly decrease the iron content by using coenzyme A or dichloroacetate here in both conditions, and also using artesunate. So this abnormal iron regulation that we observed in fibroblasts is also present in PBMC of patients. As you can see here, we have measured the iron content of PBMC grown in high iron condition. The controls here limit the iron content, whereas the patient's cells are unable to limit the iron content. And this represents a potential biomarker for future clinical trials. So to summarize that, we have here a general scheme showing the consequences of frataxin defect on iron homeostasis, and we have two types of consequences. At the level of the post-translational regulation and the post-transcriptional regulation. First of all, because the iron-sulfur cluster are needed for several functions, we observed an abnormal iron IRP-IRE regulation that results in a higher level of transferrin receptor at the cell membrane. That is the first cause of iron accumulation, of iron uptake. And also, we have a decreased transferrin ferroportin amount, and this will allow hampers the iron to exit from the cell. On the post-translational regulation, we have a decreased amount of palmitoyl-CoA because of the abnormal palmitoylation of pyruvate dehydrogenase. This is a second cause of transferrin receptor accumulation because of defective palmitoylation. Similarly, we have a decreased palmitoylation of these two antibody transporters. And I show you that we have delayed endosome recycling, and as a lot of proteins of the endosome are palmitoylated, I guess that this delay should be due to the defective palmitoylation, and this is another cause of iron accumulation. Therefore, in these cells, we have all the doors for iron uptake that are open and that cannot close, and the door for iron exit that is closed and that cannot be open. So in conclusion, fibroblasts of Friedreich ataxia have cytosolic and mitochondrial iron overload. We have shown extra mitochondrial consequences of frataxin defect, at the post-translational regulation level and also the post-translational regulation level that result in iron overload. Therefore, we can consider that mitochondria can be a signaling and regulatory organelle, at least for iron homeostasis. And finally, we have identified a new therapeutic target for Friedreich ataxia, which is transferrin receptor palmitoylation using artesunate. And before finishing, I would like to thank two very brilliant PhD students, Florian Petit and Anthony Drécourt, and I thank you for your attention.
Thank you very much, Agnès.